Everyone. This is the second virtual edition of the IIDR rounds. Thank you to our speakers for volunteering to tell us what they're doing as far as COVID research is going. So let me just introduce our four speakers for today. Our first speaker is Dr. Zain Chagla. He's an MD who specializes in internal medicine, tropical medicine, and infectious diseases. He is a consultant physician at St. Joe's Health and Hamilton Health Sciences, associate professor of medicine at McMaster University and adjunct faculty at the University of Namibia in the Department of Medicine. Dr. Chagla's research and clinical interests include global health and tropical medicine, education and antimicrobial stewardship, infections and renal transplantation, and ultrasound and infectious diseases. And he's going to talk about the hydroxychloroquine debate today, which should be interesting. Our second speaker today will be Dr. Michelle Coe. She's a physiotherapist and clinician scientist at St. Joe's, associate professor in the School of Rehabilitation Science, and holds a Canada Research Chair in Critical Care Rehab and Knowledge Translation. Canadian researchers from the Canadian Critical Care's Trial Group are contributing to the global response to understand and treat COVID-19 in hospitalized patients. And today she will discuss SPRINT-SARI, an ongoing prospective observational study, and CATCO/Solidarity, an adaptive trial. Our third speaker today is Hooman. Oh, I'm going to wreck your name, Hooman. I'm sorry. Derakhshani. Dr. Derakhshani received his Doctor of Veterinary Medicine from the University of Tehran in Iran, his MSc in Animal Science from the University of Queensland in Australia, and his PhD from the University of Manitoba. He's an NSERC postdoctoral fellow under the supervision of Dr. Surette, and his current research focuses on the application of high-throughput microbial genomics for the discovery of novel biosynthetic gene clusters and mobile elements in humans and animals. And this morning, he's going to provide an update on McMaster's contribution to the Ontario COVID-19 Genome Sequencing Initiative, summarizing the advantages and limitations of current sequencing techniques used for genomic epidemiology of SARS-CoV-2. And our last but not least speaker is Hannah Stacey, who is a PhD candidate in Dr. Miller's lab in the Department of Biochemistry and Biomedical Sciences. She did her Bachelor of Health Sciences degree at McMaster in the Biomedical Discovery and Commercialization program. And in the Miller Lab, she focuses on influenza virus immunology. But today, she's going to be presenting her work on developing assays that can be used for detecting SARS-CoV-2 antibodies from patient serum. So thank you, everyone, for volunteering to speak today. And we'll get started with Zain. So the rest of you can mute yourselves and turn off your screens.
Lori, when I'm trying to share my screen, it says host disabled attendee screen sharing.
Okay, there you go.
I'm happy to do it off the cuff, but
can you do it now?
I got it. Perfect.
All right, thanks.
1 second here.
Oh, I forgot one more second. I forgot to mention, sorry about that, that if you guys have questions, please type them in the chat box and the panelists will answer them after their talk. If they don't get to your question, I'll have them answer your question in writing. Thanks.
All right. Good morning, everyone. I'm happy to be the first present here, and thank you for organizing this. I think these are really valuable and gives us a chance to reflect on the work both of us are doing and really generate some of those collaborations which actually have started coming to fruition in a number of areas. So I really wanted to talk about this drug hydroxychloroquine, which I think finally the messaging has finally been revealed about. It's just a very interesting tale of not only a drug candidate and the evidence behind it, but also how we actually really did fail on our first attempt at finding an effective therapy for COVID-19, and really how the pyramid of evidence reversed itself going through this drug, and lessons learned for future drug exploration. Dr. Ko will be talking a little bit about some of the other drugs that have been worked out through the CATCO and Solidarity Network, including hydroxychloroquine, which is an admirable trial. But I think I really want to emphasize what happened prior and what's led to where we are today. So one conflict of interest, I was a local PI on a couple of hydroxychloroquine studies that were done, and I'll mention the results of one. They are now closed due to the non-efficacy of this drug and difficulties with recruitment. So, you know, at the beginning of this, and I remember back in January starting to read around potential drug targets, there were multiple targets that were looked at. Some of them are still being studied today, and some of them are going to be mentioned in the CATCO trial. Chloroquine and hydroxychloroquine was identified in the days of SARS-1 and as a potential inhibitor of replication. And so there was a lot of hope for this drug. It's got years of experience in terms of safety for malaria, Q fever, Whipple's disease, and more recently in the use in autoimmune diseases like lupus and rheumatoid arthritis. Because it's cheap, it's globally available. There was a lot of enthusiasm that this drug could be repurposed for COVID-19 as part of ongoing therapy. The drug works at the level of the endosome. So after uptake of the virus through the ACE2 receptor through the endosome, lysozyme is actually responsible for uncoding and then revealing the spike protein, which is involved in pathogenicity. Hydroxychloroquine actually increases the pH and decreases the efficacy of the lysosomes, which then decreases the uncoding of the spike protein and then downstream effects in terms of pathogenicity at that point. And certainly in vitro studies and cell culture studies, there was an inhibitory effect that could be replicated with human dosing of the drug. And so it's interesting, at the beginning of this, when we were starting to see what was happening in China, there was treatment guidelines that had come from China, from northern Italy, that supported the use of hydroxychloroquine in random doses, usually in the range of 400 twice a day, for moderate to severely ill patients. And you know, this was early March, late February, where we were starting to see some spread into Canada and the United States. But to that point, there was really not much data. There was one set of observational studies that was done in China of about 20 to 30 patients. There was a placebo-controlled trial of, I believe, in the less than 40-person range that's only in that was only published in Chinese, and so or in Mandarin. And so it was very difficult to actually ascertain whether or not this drug was effective, but it was being given out, you know, kind of without any real oversight for these patients. And you know, I can see the other side of the fence is that you have a critically ill patient in front of you. What else can you offer them other than the drug that you think probably has some efficacy? And, you know, if you have nothing else, why not offer it? But you know, that's not the way we do clinical research, and really we should be establishing these drugs are effective first before we start giving them indiscriminately. So this trial came out in mid to late March, which really did change the game of things in the context of COVID-19. This is by a group in France, and Didier Raoult, who's well known for his hydroxychloroquine data, had actually derived some of the data around Whipple's disease and Q fever, and is, if you can look at the last name, also the namesake of the bacteria Rallotella, who generated this study. It was a very poorly constructed study in terms of methodology. They really just took a bunch of patients that were admitted to their institution and gave them hydroxychloroquine and saw their viral loads drop. There was no real clinical data around these patients. And then in 5 of those patients, they gave them azithromycin on top of it, which is an antimicrobial that targets the ribosome and has anti-inflammatory properties. And on top of that, just saw that, oh, there was a bigger drop. And the reason for the azithromycin wasn't necessarily the use for the viral infection, is actually to prevent bacterial superinfection. And so the control group in this sense was a number of patients that were admitted to other institutions that they just had data on. Again, there wasn't a lot of clinical data on these people other than that a significant amount were actually asymptomatic and a small percentage were symptomatic with some changes. These were not ICU patients. But the data he presented showed that there was a reduction in PCR positivity in the folks that got hydroxychloroquine as opposed to placebo. And then in the people that got azithro, that again, the 5 patients that their conversion to PCR negative on nasopharyngeal swab was even more remarkable. So this is just if they were positive or negative at any given time. And you see the dichotomy here. This is the actual data set from the study. And you can already start seeing the pieces starting to fall apart here. So for example, the top group here is the control group. A lot of the days of reassessment for these studies, ND is not done. They were counted as positives. And we know now lots of people can flip back between positive to negative, and whether or not that has any statistical significance for a test that's really just looking at or clinical significance, a test that's looking at nucleic acid is variable. You also see cycle thresholds at the higher point here as compared to some of the higher point cycle thresholds here. And so whether or not that's even clinically relevant, do they have just dead or decaying virus, is also questionable at that point. So, the control group, although he counted a lot of them as positives, could have very well been negatives. There were only 20 patients enrolled. And then there's another random 6 patients here that were lost to follow-up, either that they were discharged or they were in the ICU. And 5 of them were patients that did receive hydroxychloroquine as part of their therapy. They weren't counted in this analysis of only 20 patients. And a number of them were actually positive at the time of their dropout from the study. So they weren't even counted in this analysis. So, very poorly designed study. And unfortunately, the wrong person got a hold of this study and decided to take it to the next level. So this is Donald Trump going through this. And really, this led to a huge issue with stockouts and drug stockout costs. And interestingly, as this progressed, there were many more observational studies that came out saying that hydroxychloroquine was not effective and actually had a mortality yield. For those of us that were actually doing these clinical trials, those observational trials then created Health Canada and FDA warnings that made us go back and justify why we were doing a randomized clinical trial on this drug, which is, again, completely opposite to the pyramid of evidence here. This study emphasized the issue with the observational data. So this was a registry analysis by Sapp and Desai and the Surgisphere group, which many of you may have heard of, essentially looking and noting that hydroxychloroquine actually had a 3 times higher mortality than placebo or non-hydroxychloroquine in the treatment of patients. Essentially, this trial was actually retracted when there were huge issues with the data it presented. The Australia data, for example, that they used actually quoted in the 5 centers that they looked at this more mortality than the entire country of Australia. And a lot of scientists actually took down this study, pointing out the flaws in this data and a similarly linked New England data set as well. Finally, this is one of the trials actually that we were involved in that actually came out with randomized control data looking at post-exposure prophylaxis of hydroxychloroquine and showed no difference in people with who had got hydroxychloroquine immediately after their exposure to COVID-19 versus placebo. I won't go into the technique of this study other than to say it was very unique in the sense that we used a model where people could register online. And then the recovery trial, which is probably the one that has generated the most data to date about clinical trials, has found no difference in hydroxychloroquine as compared to placebo. And this data actually has essentially cut off hydroxychloroquine from a number of other large ongoing clinical studies. And unfortunately, another study, which only came out actually a couple of days ago, of an observational study that was done at Henry Ford, seems to show hydroxychloroquine has an effect. When you actually look at the subgroup analysis, it's actually tied to a lot of the steroids that were used in this study. But again, a certain individual seems to have doubled down on this drug, and it really has become a Democratic Republican issue, which is just incredibly unethical in the context of this pandemic. So that's it. I think I'm running out of time here. I just really wanted to present this story to say, you know, there are really well done clinical trials that are ongoing, but this hydroxychloroquine, the answer was actually bombarded by poorly done clinical trials and observational trials based on a single poorly constructed trial to start with. The lesson has been learned. I think the further trials that are ongoing, we are going to wait for randomized clinical data rather than going back to what we did with hydroxychloroquine. But certainly this is a dark lesson as part of the beginnings of this pandemic clinical trial strategy. And with that, I will stop sharing my screen here. I don't think there's any questions in the Q&A.
Thank you.
I'm happy to answer at the end.
Has anyone done a study just of these drugs like azithromycin that have anti-inflammatory properties without the hydroxychloroquine?
Yeah, there are a couple that are enrolling. They haven't actually gotten to full treatment. I think recovery actually may have an arm, but I'm not 100% certain if azithromycin is part of it. And then Michelle, who's a part of Remap CAP, which is one of our other studies, looking at different domains for pneumonia, including COVID pneumonia, has an arm for azithromycin as part of treatment alone. So that will also be interesting data in that context.
And I know a lot of the hydroxychloroquine studies were done in severely ill patients, and you know there's some thought that maybe it's just too late for the drug. Yeah. So were the trials designed to be more prophylactic in nature, or is this
so that first post-exposure, the Bullaware trial from the New England Journal, was immediate, and that we participated in, was immediately after diagnosis. You were shipped the drug within 5 days, and that showed no difference between people who got it versus placebo. We have a second trial with that same protocol that was immediately after diagnosis to be given the drug to start within 5 days of diagnosis for non-hospitalized individuals, and that data set we're just going through the data analysis now.
Okay. Any questions from the audience? If you have a question, please type it in the chat. Okay, I'm not seeing any questions. So, oh, wait a second. I spoke too soon.
Apparently, Bolsonaro is taking hydroxychloroquine. Apparently, he has COVID-19 too, so it didn't work, clearly.
I noticed he's wearing a mask now, though. So, you know, little steps. They're modeling the behavior. Okay, thank you very much, Zain. I think we'll move on to Michelle. So Michelle, you should be able to share your screen now. And you'll need to unmute yourself. Okay, Michelle, you should be
Thank you. Thanks so much. All of the technologies.
Yeah.
All right. Well, thanks for the opportunity to share some of our work, and I appreciate the chance to share about what we're doing as part of what we call Team Canada in COVID-19 research. I'll speak today about both SPRINT-SARI and CATCO, and it's been lovely to learn about the ID world as part of this pandemic. I'm really grateful to my colleagues, the Canadian Critical Care Trials Group, who have really been instrumental in helping to bring some of these studies to Hamilton. So you might be wondering, how does a physiotherapist become involved in COVID-19 research? Well, a lot of it was because of the Canadian Critical Care Trials Group. And for those of you who aren't familiar, This is a group that was established in 1989. It has important ties to McMaster through Dr. Deborah Cook, who was one of the founding members. And its mission is to advance the care and improve outcomes of critically ill adults and children through research. We have over 350 publications, including several in the New England Journal. And one of the things that you're learning about now about COVID is that survivors of the ICU have important disabilities. And our group has led a landmark study in survivors of ARDS and also in SARS to understand what patients and survivors might expect after surviving their hospital stay. So briefly, I'm going to speak to you about two studies. One, which is SPRINT-SARI, which discusses the epidemiology of COVID-19. And second is CATCO, which is a treatment study for COVID-19. When the pandemic broke out, it was critical that we tried to understand more about the epidemiology of disease. And wouldn't it be great to have research studies that could do so? But as all of us know as scientists, it takes time to carefully plan these studies. And as Zain mentioned, it was important to try to understand how these different studies work and how to understand the disease. And in fact, SPRINT-SARI was perfectly timed as a sleeper protocol to understand the epidemiology of COVID. This is a sleeper protocol that was in place, led by Dr. Surette Murthy, who is a pediatric ICU doc, but also ID specialist. And this study was actually published just last year to show the feasibility of conducting a multi-center, observational, prospective study of severe acute respiratory infection. SPRINT-SARI itself includes both adults and children. It looks at different parts of the disease, including the incidence, disease severity, different risk factors, case fatality, length of stay, microbiology, and also the treatments that were received during hospitalization. And here in this timeline, you can see the tremendous speed at which SPRINT-SARI has ramped up. Anyone who has been involved in doing multi-center trials or multi-center studies understands the challenges in starting to ramp up studies very quickly. By April of this year, almost 6,000 patients were enrolled. And by June, over 42,000 patients in almost 500 sites and 37 countries have contributed to this global data set. There's both a rapid and full CRF or case report form available. There are add-on modules in development. There are dashboard updates. And one of the wonderful things about SPRINT-SARI is that it's open access, it's a global data set, and people can request access through a data access committee. However, there is prioritization to collaborators from participating sites. I'll just give you a flavor of the different types of data that were available in the most recent data release. So here you can get a sense of some of the patient characteristics. In the bottom right-hand corner, there's the website that you can access to get a sense of the larger data report. Information of the different types of symptoms that patients presented with, recognizing that these are hospitalized patients, and also some of the comorbidities that patients have presented with at hospitalization. We'll highlight that this is a center that is coordinated in Canada at Sunnybrook. The leads are Surette Murthy and Rob Fowler. We currently have 53 sites actively enrolling in Canada, and we have an ICU paper in progress. I also wanted to share with you the people who are also behind the scenes in collecting the data at St. Joe's, Laurel Kelly and Heather O'Grady, And this is a study that's also activated at McMaster Children's. If you as an investigator are interested in participating in SPRINT-SARI, here are the contact details for Ashgar Rishu at Sunnybrook, and I'll propose this also at the end of the talk. Next, I'll briefly give you an overview of CATCO. And CATCO is our Canadian contribution to the WHO Solidarity Trial. And as you know, there were several people, many groups around the world that became interested in helping us try to find treatments to improve outcomes in patients with COVID. However, we also know that multiple trials, small little efforts around the world are not very helpful. And Zain identified the challenges, particularly with some of the initial trials with hydroxychloroquine. So what the WHO Solidarity Trial is a global mega trial. Its goal is grand, to enroll thousands of patients with a common analytic set. It targets hospitalized adults with COVID, and they were randomized initially to 5 arms. And as they mentioned earlier, hydroxychloroquine was discontinued, but so was Kaletra. What's really interesting about this trial is that it's an adaptive trial. There's no statistical analysis plan, which is counter to anything that you would learn in school. There are no pre-specified stopping rules, but there's no sample size calculations. But as an adaptive randomized trial, one of the benefits of this new methodology is that we can look at different treatments concurrently with a DSMB or safety monitoring board and identify when treatments are starting to not be effective. Briefly, these are the eligibility criteria for those who can get into the trial. We're looking for adults with a laboratory confirmed SARS-CoV-2 infection and are also hospitalized at a participating center. Patients excluded from the trial are anticipated to be transferred to another hospital, not expected to survive, have known allergies to study medications, or are also receiving one of the study drugs at the time of enrollment. This study is also coordinated through Sunnybrook. It is, the different treatment arms that are part of CATCO include optimized standard of care, Kaletra or the lopinavir-ritonavir, hydroxychloroquine, and also remdesivir. You can see this timeline that shows the availability of the different arms of the trial. You'll notice that remdesivir has also been in the news, and we have been actively working to get remdesivir available for us in Hamilton. Right now, the only 2 arms that are open in CATCO include the optimized standard of care and remdesivir as soon as it's available. Finally, CATCO is coordinated through Sunnybrook. Again, it's funded by CIHR, and remarkably, since the initiation of the pandemic, we are currently actively rolling in 35 sites. There are 20 sites also in progress. And I'll also highlight the local leads with Dr. Eric Dwan and Jason Chung and France Clark. So I'll close by giving you this overview of both SPRINT-SARI and CATCO. And if your sites are interested in contributing to either of these efforts, these are the contact information details. Thank you.
Thank you very much, Michelle. You can stop sharing your screen and we can entertain questions from the audience. I have a quick question for you while we're waiting for people to type. So the remdesivir is looking good, but I heard that basically the US has bought up the entire supply until at least September. So what's the plan to get a hold of this material?
So we've been, so my understanding is that we've been in active negotiations with the company, and we are working at securing access to remdesivir as part of CATCO. So those negotiations are ongoing, and we anticipate access to the drug.
Is it possible to spread the manufacturing out, or is Gilead basically not letting anyone else make it?
That's a very good question. Zain, I'm not sure if you're more aware of the access to remdesivir.
Yeah, right now it is a Gilead drug. There is a generic company in India called Dr. Reddy, which apparently is looking at trying to make a generic form of remdesivir to distribute to the rest of the world. But for typical use, it is only restricted to the United States for now.
Okay, there's a question for Michelle from Matt Miller. Dexamethasone has shown very promising results recently. Any thoughts of adding it?
That's a really good question. That's something that I would need to find out from our investigators.
Yeah, I can add a little bit to that. Is that unfortunately, I think that the data, or fortunately, I think the data from Dex is probably putting it towards the standard of care for oxygenated patients. So it would be now probably ethically very difficult to actually randomize people to a non-dexamethasone based arm as part of their optimized standard of care.
And I've read some stuff online about turning the patients onto their stomachs to help them breathe.
Yes, proning.
Yes. And what's the rationale with that?
So physiologically, patients who present in the ICU typically present with something called ARDS, or acute respiratory distress syndrome. And the thought is that by proning patients, that will improve their ventilation and perfusion matching.
Jerry, did you have a question?
Yeah, I do. Thanks. I can't type now that you've made me a participant or whatever it is. I am mystified as to why Kaletra is on the list. It's a mixture of aspartyl protease inhibitors for HIV. There are no aspartyl proteases in coronaviruses. They're all cysteine-based proteases. What's the thinking there?
Zain, I'm actually going to punt that question over to you. Not sure if Zain's available.
I was on mute, sorry.
Mute.
There was data of lopinavir ritonavir in SARS, and it had been tried also in the MERS coronavirus. There was some clinical response, although it's not really clear how effective it actually was. And so that's why it got tossed into this mess. But I agree with you, like the mechanism didn't seem favorable, but it's, there was in vitro data in SARS and MERS to confirm its potential use.
Okay, thanks.
All right, thank you. We had one additional question from Jake Magdalene. Is international coordination to minimize redundancy between trials going on?
Yes, there is. And I think that's been one of the wonderful things about this adaptive trial and also the WHO, because they are working with different groups to actively collaborate and coordinate. And the discontinuation of the hydroxychloroquine arm was as a direct result of collaboration with other groups. So you can also look at this randomized adaptive trial by allowing the groups to work together, pool their data, and quickly come up with answers so that we can also pursue other treatments and bring those into the randomized trial.
Great. All right, I think in the interest of time, we'll say thank you very much, Michelle, and we'll move on to Hooman. So Hooman, you should be able to now share your screen. Looking good.
Can you hear me?
All good.
Can you hear me?
Yes.
Okay, great. So today I'm going to give you an update on McMaster's contribution to Canadian COVID Genomic Network. And I would also discuss with you some of the challenges of sequencing techniques that we use for sequencing the genome of SARS-CoV-2. So back in April 2020, the federal government announced $40 million investment to support the Canadian COVID Genomic Network. So of this money, about $20 million would go towards sequencing of up to 150,000 SARS-CoV-2 genomes, and the other $20 million would support sequencing of up to 10,000 genomes of the Canadian patients. The overall goals of this project is to first create a virus patient data bank that could help inform decision-making by a public health agency of the Canada. The next goal is to create capacity to address both the current and the future viral outbreaks in Canada, and this includes capacity for sequencing a large number of viral and human genomes, and also the computational resources that are required for processing the large genomic data. And the last goal is to create a framework that will facilitate data sharing both nationally and internationally, and the main reason here is to promote collaborations. So in Ontario, a coalition of public health laboratories and research institutes, including McMaster University and Ontario Institute for Cancer Research, they are mainly responsible for genome sequencing. Among these, McMaster sequencing territory covers a relatively large area, with samples being shipped to us from research hospitals in Ottawa, from Sunnybrook and Sinai Health Systems in Toronto, and of course, St. Joe's and Hamilton Health Centers. They're all sending the samples to McMaster's genomic facility for sequencing. But what can we learn from sequencing the viral genome? The first and most important application is to use the genomic data to track mutation and evolution of the virus over the time. So on this global phylogenetic analysis of the SARS-CoV-2 genomes, we can see that the genome of the virus, which originally initiated in Wuhan, China, has been constantly mutating and evolving into new lineages. At each of those lineages, at the moment, they are spreading in different parts of the world. By projecting the source and relationship of this genome on a global map, we could also get information about the transmission routes of the virus. And by knowing that, we could implement strategies that we can limit the spread of the virus by those. The other application of the genomic data is to identify evolutionary conserved regions of the virus genome. Here is an overview of the SARS-CoV-2 genome. And on the top, the bars shows the frequency of the mutations that have been so far identified at each position of the genome. So we can see certain mutations, such as this one on the spike protein, they're happening at a higher frequency compared to the rest of the genome. And on the other hand, we have part of the genome that they are pretty much conserved and they don't experience that much mutations. From the perspective of development of new vaccine and treatments, ideally we want to target antigens that are coded by the conserved regions so that we can avoid frequent development of resistant variants. The other application of this data is to help us design and improve the diagnostic PCR assays. And here, we want to guide away our primers from these variable regions and focus them on the more conserved regions of the genome. So we can reduce the rate of false negative tests that usually happens due to the mutations at the primer binding site. So there are currently three main approaches for sequencing the SARS-CoV-2 genome. The first one is total RNA sequencing, or what we refer to as direct metagenomics. Here, we extract and purify total RNA from clinical samples. We then reverse transcribe it to total cDNA. And without any further enrichment of the viral cDNA, we prepare our sequencing libraries. The main advantage of this approach is that because we are using sequencing of the total RNA, it enables us to characterize the genome of the novel pathogens. So it is very useful at the onset of the viral outbreak when we want to characterize the causative virus that is causing the outbreak. It also enables us to detect co-infections with bacteria and fungi that might be present in the clinical samples. And by using the human mRNA data, we could also characterize post-transcriptome, such as changes in the expression of the immune response pathways. The main limitation of this approach, however, is that it has very low sensitivity to detect viral genome. And the reason for that is the dominance of human RNA, which accounts for more than 99% of the total RNA. So in order to achieve a decent coverage of the viral genome, we need to perform ultra deep sequencing, which makes it pretty expensive and obviously not applicable for large data sets. The second approach is target enrichment sequencing, or what we refer to as probe capture enrichment. Here, we have a set of genome-specific probes that are usually attached to magnetic beads, and we use these probes to capture and enrich the viral genome materials. We could either add these probes directly to the cDNA in order to capture and pull up the viral cDNA, and then use that cDNA to prepare our sequencing libraries. Or we can first prepare our sequencing libraries and then add the probes in order to enrich the libraries that belong to the virus and then sequence those. Either way, using these probes would enable us to significantly increase the coverage of the viral genomes. And by doing that, we are reducing the sequencing cost per genome, which is really desirable. And the other benefit of this approach is that by designing probes that they can target multiple pathogens at the same time, we could also detect co-infections with other viruses and bacteria that are included in our probe sets. But the limitation of this approach is that since we need to design probes, genome-specific probes, we need to have access to a decent genome assembly of the target pathogen. So it is not applicable for sequencing the unknown pathogens. And the next limitation is that the efficiency of probe hybridization usually decreases at the lower concentration of the RNA. And with clinical samples, where we have a lot of samples that have low RNA concentration, we could end up with incomplete coverage of the genome, which is not ideal. And the last but most widely used approach is PCR amplicon sequencing. So in this approach, we use a set of overlapping PCR primers to amplify the entire genome of the virus. Then we can use the generated PCR amplicons to prepare our sequencing libraries. The main advantage of this approach is that it has the highest sensitivity for detection of viral genomes. It could work with as low as 10 viral RNA copy number in a single reaction. And as a result, it offers us the lowest sequencing cost per genome. But the limitation of this approach is that, similar to probes, because we need to design primers, we need to have access to a high quality reference genomes. And this makes it not applicable for unknown pathogens. And the other problem is that for certain amplicons, we would have loss of coverage, or what we call amplicon drop-loss. And this is usually due to mutations at the primer binding site. And the other reason that could result in loss of coverage is also a low integrity of the input viral RNA, which is also typical for clinical samples. At the moment, there are many primer sets available for amplification of the SARS-CoV-2 genomes. One of the most popular and widely used primer set is the Arctic primer set. Here we target 400 base pair amplicons, and it is composed of 98 different primer pairs. The other primer set is Eden primer set, which targets 2.5 to 5 kb amplicons. But the limitation of amplification of large fragment is that these primer sets are usually very sensitive to the integrity of the input RNA. For example, when we tested the Eden primer set using the high quality RNA that we isolated from cell culture samples, we were able to successfully amplify 5 kb amplicons. But when we tested the same primer set with clinical samples, we couldn't get anything. And the issue here is the harsh chemical and high temperatures that the clinical labs are using to inactivate the virus. And this results in degradation of the input RNA. So we switched to the Arctic primer sets, and fortunately, the Arctic primer set provides us a decent coverage across the genome of the virus. But the issue with the Arctic primer set is that the complexity of the PCR reaction, because we are combining so many primers in a single PCR reaction. Some of these primers, they have lower efficiency compared to the other regions. So as you can see here, although we have an average high coverage of the genome, for some fragments, we have drop in the coverage. And this problem becomes more severe with decreasing viral RNA concentrations. So we have been working to address this issue by target enrichment of the input cDNA. So we are trying to use some capture or primer-based enrichment of the cDNA right at the reverse transcription step to increase the concentration of the cDNA that belongs to those low coverage fragments. And finally, I would like to give you an example of the results that we have generated so far. So this is a phylogenetic analysis of the initial 300 samples that we received from Sunnybrook, and this is a collaborative study with Dr. Mubaraka from Sunnybrook and also Dr. MacArthur. So on the heat map here, we can see the frequency of the mutations compared to the reference Wuhan genome. So on the sequence genomes that we included in this analysis, we were able to detect two frequent and dominant mutations that are shared among all the genomes that we have sequenced. And there is growing evidence that these mutations are changing the behavior of the virus. Like this paper, which shows that the mutation in the spike protein amino acid position 614 is increasing the infectivity of the virus. So there are many exciting things that we can learn from this data as we move forward. And as of today, we have sequenced around 1,000 viral genomes in the Surette lab and the Farncombe Genomic facility. This is only possible through the tremendous support that I received from Dr. Surette and also the help that our amazing technician, Tori Markov, has provided to us. And I would like to finish my talk with mentioning that this work has been an amazing collaboration among different labs across the country. So we received tremendous support from the members of Surette's lab and also the Farncombe Genomic facility. Dr. MacArthur's lab and his collaborators, they are working tirelessly to develop new bioinformatic tools in order to process the huge amount of data that is being processed nationally. And of course, we have our clinician collaborators who are working around the clock to process hundreds of samples on a daily basis. So thank you all for listening, and I would be happy to take any questions.
Thank you very much, Hooman. We have a couple questions, both from Desmond Hamill. He says there are no sequences published in GenBank from Canadian isolates. However, there are plenty from other countries such as the US, Italy, etc. Can you elaborate why? And is this not within the mandate of Health Canada or PHAC, or are Canadian sequences only coming from academic institutions?
So there is another website that researchers, they are on a real-time basis uploading their sequencing data. So this is specific to virus sequencing data. So it's, if I'm not wrong, it is GISDA. So this is the website that they are uploading. So it wouldn't be necessarily uploaded to the NCBI. And in Canada, so this initiative is pretty new. So we haven't yet published this data. Of course, Dr. Andrew MacArthur will comment better on this, topic, but I'm sure that the data would be uploaded as we move forward. Yeah. Okay. And the mutation that you showed in the spike protein, this hypervariable point, is that the same D to G mutation that's been reported elsewhere? That's a very famous widespread mutation that is reported to be dominating all around the world. Yeah. And there hasn't been any other changes at the same position to other residues? Not at the same position, but that mutation is co-occurring with 3 other mutations across the genome. So those mutations, one of them is for the RNA-dependent RNA polymerase, and the other one is for non-structural protein number 2, which they don't know what's the function of that one. So these mutations, they are occurring at the same time. But and is the general sense that this is making the virus more or less infectious? So it's, I mean, the recent paper that I showed you, it just came up 3-4 days ago. So in that paper, they have some in vitro experiment that shows that it is increasing the infectivity, but it doesn't necessarily change the outcome of the disease. So it's a very controversial, and I think we need more data to validate this one. Okay, thank you. I'm not seeing any other questions. I'll give it one more 10-second pause, just because I know some people type slower than others. All right, if you have any other questions for Hooman, you can type them in the question and answer or the chat, and he can answer them offline. So thank you very much, Hooman. And thank you for working so hard on this. Okay,
We're moving to our last speaker, who is Stacey. Hannah, you should be able to share your screen now.
Okay. Okay, so good morning, everyone. Today I'm going to talk about a few different quantitative and functional serological assays for detection of antibodies against SARS-CoV-2. So as this global pandemic has continued to progress, our understanding of the antibody response to this virus has also deepened. So we now understand that individuals who become infected with this virus mount an antigen-specific IgM response, followed by an increase in antigen-specific IgG and neutralizing antibodies. And we also see a correlation with the severity of clinical disease and the magnitude of this antibody response. So individuals who have worsened disease outcome often have higher titers of these antibodies, whereas individuals who are asymptomatic or have less severe disease have lower titers. And there's been reports that individuals who are in this asymptomatic region of the protein. And so having antibodies that interaction are a really effective way to effectively reduce the entry of the virus into host cells. However, I do want to mention that just because a lot of the attention has been focused on spike antibodies, it doesn't mean that the immune system can't see other viral antigens and proteins. In fact, the immune system can generate antibody responses to a variety of other structural and non-structural proteins. And one really good example of this is with the N protein, which is the most abundant protein in the virus. However, because this protein is an internal protein, antibody responses targeted here probably are not protective. And it has been demonstrated that this is probably a better antigen for T cell responses. So the focus has remained on inducing spike-specific antibodies. And the quantification of these antibody responses are beneficial for a number of reasons. So not only are they beneficial for identifying patients who have experienced prior SARS-CoV-2 infections, but they can also be beneficial for identifying patients that are good matches for convalescent serodonation and looking at the efficacy of potential therapeutics like monoclonal antibodies and single-chain antibodies. And one of the best tools at our disposal right now for quantification of these antibodies is the use of an enzyme-linked immunosorbent assay, or an ELISA, that uses recombinant receptor binding domain and full-length spike protein. And really nice detailed protocols of this method have been published in Nature Med and current protocols in microbiology recently by Dr. Florian Kramer from the Icahn School of Medicine in New York. So the way that these assays work is that you're able to generate recombinant protein using a mammalian cell expression system, and we get really high yields of this protein using this method. And then you can coat a 96 well plate with these recombinant proteins, followed by the addition of serial dilutions of your patient sera. And then you can come in with a probed secondary antibody that's specific for a variety of different isotypes of human antibodies. So you can detect IgM, IgG, IgA that is antigen specific. And using this method, you're able to relatively quickly, in a matter of hours, detect a magnitude of response for SARS-CoV-2 specific antibodies. And in our hands, this worked really well at being able to distinguish between individuals who had prior SARS-CoV-2 infection versus those who did not. So both the recombinant RBD ELISA and the full-length spike ELISA were really effective at showing who had these antigen-specific antibodies. One thing to note here with these experiments is that the recombinant RBD ELISA is very highly specific. So when they were optimizing these assays, even individuals who were swab confirmed positive for other seasonal coronaviruses like NL63 didn't show cross-reactivity to the receptor binding domain. Whereas the full-length spike ELISA, you do lose some specificity just due to the increase in epitopes that are present on the full-length protein. Additionally to the ELISA, there has been reports of these rapid serological detection methods. So these are what's being proposed for immunity passports. And one way to do this is the lateral flow detection method. And the biochemistry of these assays is actually very similar to the ELISA. However, with these assays, you're really only able to see a binary yes or no answer, whether your sample has the antigen-specific antibodies that you're looking for. So you get 2 lines if it's positive and 1 line if it's negative. And while both of these methods are effective at determining antigen-specific antibodies, it's important to note that there are pros and cons to these methods, and that you should keep these in mind when looking at what your question is and what types of information you want to gain from doing these experiments. So something like the lateral flow detection methods are really good for high throughput, really fast, low cost detection, but they aren't going to tell you whether the magnitude of the response is high or low, which can make inferences about protection very difficult. Whereas doing a quantitative ELISA, which does take a little bit more cost and a little bit more time, does give you more information about the magnitude of the response in your samples. And then when coupled with more functional assays, like a microneutralization assay, you can really start to be able to establish correlates of protection, which will become important when we're starting to look at vaccine responses. So when we're looking at the microneutralization assay, which determines antibody function, not just antibody specificity like an ELISA, one hurdle that we faced is that working with wild-type SARS-CoV-2, you're limited to the level 3 facility. So doing high throughput analysis of serum is a little bit cumbersome. So we had to overcome this by developing pseudoparticles using a lentiviral packaging system so we could do these experiments at level 2. So we generated these pseudoparticles that express full-length spike on the surface of the virus, as well as a luciferase gene. So when the pseudoparticles get into cells, the luciferase is expressed, and we can read out the assay with a luciferase reagent. So if a patient has anti-spike antibodies, they bind to the pseudoparticles and prevent luciferase expression, and we can use this reduction of expression as a readout for neutralization. So keeping this in mind, the way this experiment works is that we have our SARS-CoV-2 serum or patient serum of an unknown infectious background, and you can in fact inactivate it at 56 degrees. Then you serially dilute it and incubate it with the pseudoparticles for 1 hour at 37. And then you can add the immune complexes onto a lung epithelial cell line that expresses ACE2. And following an 8-hour incubation, we add luciferase reagent and we read this out on a plate reader. So far we've done this experiment using some sample COVID donor serum, and we can see that there are in fact neutralizing antibody responses in a variety of these patients, although the magnitude of this response varies greatly on a patient-to-patient basis. And moving forward with these assays, what we really want to be able to do is correlate the magnitude of antibody responses that are seen with ELISAs to the neutralizing antibody responses that we see with the MNTs. And when we couple these things together, what we'll really be able to do is establish strong correlates of protection, because right now those really aren't well recognized for SARS-CoV-2, and there's vaccines that are in clinical trials in phase 3 that are moving forward without any correlates of protection really being established. So when those have been established, you'll be able to look at the immunogenicity data and know more early in your clinical trials if you're reaching protective antibody levels, which will be excellent moving forward. So just to kind of summarize, we have a variety of tools for quantifying antibodies at our fingertips right now, but there are pros and cons to both of these that need to be taken under consideration when choosing these methods. And I just want to acknowledge my supervisor, Dr. Matthew Miller, our amazing lab manager, Jan Eng, who's been my partner in crime in optimizing all these assays from the start, and some of our collaborators. And I'm happy to answer any questions.
Thank you, Hannah. That was really great. I have a quick question for you. So I've heard that, if you don't have severe disease, you don't really mount much of an antibody response to this virus. So the patient samples that you're getting, are those from severely ill patients or a whole spectrum?
So we've just started working with the collaborator now, and we'll be looking at ELISA and MNT titers from patients in different phases of disease. So acute phase, ICU phase, and convalescent phase. So we're going to be able to answer soon whether the magnitude of the response is different at different phases of disease. The ones used for these assays were all recovered patients. So
that was sort of the second part of my question is, what's the duration of the response? Because it doesn't look like it's great right now from what I've heard.
Yeah. So I think we have to go off of kind of historical data with the original SARS-1 virus. So we know that people who had that virus and were infected, people saw antibody titers as far out as 2 to 3 years, but those were people who were bona fide infected with the virus. For this virus, it has been suggested that if you were asymptomatic, your antibody response is lower, and they have already noticed waning of those titers.
Okay, so Des says, truly excellent rounds, great mix of clinical and benchtop. Thank you all speakers. And Eric would like to know, is the vaccine going to work? That's a pretty...
Oh, that's heavy. I don't know if I can... I hope.
Don't we all?
So, Lori, I have a question, which is actually kind of relevant to what Eric's asking. I think I'm obsessed with testing, and I guess the question I have is the fast the pregnancy test kind of test that you're describing. What are our chances of getting something like that to work super fast and just using something like saliva, which is easy to do, so that we can test and test like crazy to isolate people before they're symptomatic?
I think the chances are really good. I think it's just setting it up in a way that we know that the information we're getting from those binary assays correlates really well with something like a quantitative ELISA. So in parallel to doing testing with the binary kind of pregnancy test method, you're going back and checking to make sure that those are true positives every 100 of samples, or when you're setting up your threshold with the binary experiment. Those binary assays also are really good when you're doing seroprevalence studies in areas where there's already high prevalence of infections. So if you are losing a little bit of specificity, then it doesn't impact your results as much.
Yeah, it's my understanding too that you won't, this would not be a screening method for the asymptomatic period, because you wouldn't have
of course
antibodies yet, right?
Yeah, no, that's Matt's point. I guess I'm just obsessed. I'm thinking of, so the Venter Institute, for example, is back to work, and they're testing everyone in their institute once a week.
Wow.
For virus.
For virus.
Yes.
Yeah. So, and I think that provides a level of safety that you could get ahead of this and not just react what you get infected. So I'm trying to figure out how we can stay ahead of the curve here as an institute. Andy, you need to save us.
Wear a mask. Wash your hands. Watch your distance. All right. Matt says, yes, there will be a vaccine that works, and that antibody tests are probably not useful to identify patients before symptom onset. I think everybody loves the idea of immunity passports, but I think that's a tricky concept, especially if immunity doesn't last as long as we hope. All right, in the interest of time, because I see we're after 9 o'clock now, I just want to thank all of our speakers again and give you an applaud. It's hard to give a talk when you're not getting any feedback from your audience, but I commend you on your presentations, and thank you very much for telling us what you're up to. Thanks everybody for joining us.